Bismillahirrahmanirrahim. Dear students, have a nice day. Hope you will be fine, best of your health. This is Dr. Sajid Mahmood, Division of Science and Technology, University of Education, Lahore. Today we are talking about steroid hormones, steroidogenesis, its applications and biosynthesis. This is our second lecture on steroidal hormones, in which we will discuss the synthesis of corticoids, cortisol and aldosterone. We will also discuss how different kinds of hormones are derived from the cholesterol, which is the basic precursors, as well as we will discuss the functions of different kinds of steroidal hormones. Let's see the synthesis of corticoids. Corticoids are divided into gluco and mineralocorticoids hormones. The predominant human glucocorticoid, cortisol, is synthesized in the zona fasciculata of the adrenal cortex. The synthesis of cortisol involves hydroxylations of progesterone at the 17 alpha carbon 21, that is represented by CYP21 and 11B. CYP11B1 are the positions which are mentioned upon carbon 21 progesterones. The synthesis of cortisol is under the control of an anterior pituitary hormones, ACTH, and negative feedback mechanism in which elevated cortisol suppresses the release of ACTH. The dominant human mineralocorticoid is aldosterone, which is produced in the zona glomerulosa of the adrenal. The synthesis of aldosterone involves the synthesis of corticosterone and subsequent hydroxylations and oxidations at carbon 18 to yield aldosterones. The synthesis of aldosterone is regulated directly by levels of potassium and indirectly by the effect of sodium levels and blood volume on levels of angiotensin II. Synthesis of cortisol. Cortisol is the example of corticoids. Let's see how cortisol is synthesized. In the first step, pregnenolone is converted into 17-hydroxy pregnenolone in the presence of CYP17 alpha hydroxylase enzyme. You can see in the first reaction the pregnenolone, in which the acetyl group is present at carbon 17 position, is converted into 17 hydroxy pregnenolone in the presence of enzyme CYP17. In the second molecule, there is a presence of acetyl group at carbon 17 position in addition with hydroxyl group at carbon 17 position too. So in the next step, the 17 hydroxy pregnenolone is converted into 17 hydroxy progesterone in the presence of 13 BHDS enzyme, which is then converted into deoxycortisol in the presence of 21 hydroxylase enzyme. You can see in the second step of reaction, 3-BHSD enzyme affects the 17 hydroxy pregnenolone, which is converted into 17 hydroxy progesterone. In the progesterone, you can see over here in this structure, there is a presence of acetyl group and hydroxyl group at carbon 13 positions, while acetyl group at carbon 17 position. So this molecule is further converted into 11-deoxycortisol. In deoxycortisol, at carbon 3 position, there is a presence of carbonyl group, and at carbon 17 position, there is a presence of methoxy group addition with the carbonyl group. Further, 11-deoxycortisol is converted into cortisol in the presence of 17 beta hydroxylase enzyme. In the reactions over here, you can see 11-deoxycortisol is converted into cortisol in the presence of CYP11B1 enzyme. The difference between both the structure is that There is a presence of hydroxyl group in the cortisol at carbon 11 position and further hydroxyl group at carbon 17 positions, in addition with methoxy group which is attached to the carbonyl group at carbon 17 position. There is also a presence of double bond or unsaturation at carbon 4 position in the cortisol. So, in both the structures, the difference between the function moieties is at carbon 11 position, carbon 4 positions and carbon 17 position. So, this is the method for the preparations of cortisol. Synthesis of aldosterone. Aldosterone is synthesized in the body from corticosterone, a steroid derived from cholesterol. The production of aldosterone in adult humans about 20 to 200 micrograms per day in the zona glomerulosa of the adrenal cortex is regulatory by the renin-angiotensin system. So, in the first step, the cholesterol is converted in pregnenolone in the presence of a specific enzyme. In next step, the pregnenolone is converted into progesterone in the presence of 3-BHSD enzyme, which is generally represented as 3-beta hydroxysteroid dehydrogenase. After that, progesterone is further converted into deoxycorticosterone in the presence of CYP21A2 enzyme, that is written as CYP21A2 hydroxylase. At the end, deoxycorticosterone is converted into corticosterone in the presence of CYP11B1 beta hydroxylase enzymes, and then it is converted into aldosterone, which is our target compound, in the presence of CYP11B2 aldosterone synthase enzyme. Let's see the synthesis of different hormones from cholesterol. First of all, we will discuss the structure of cholesterol. The total number of carbon atoms of cholesterol consists of carbon 27 atoms, in which the hydroxyl group is present at carbon 3 and double bond is present at carbon 5. The hydrocarbon side chain, which consists of total number of 7 carbon atoms, is attached at carbon 17, in which two methyl group are present at carbon 25 and carbon 20 positions. In addition, two extra methyls are present at carbon 18 and carbon 19. So, this is the common structure of cholesterol, which is generally known as cholest 5-ene 3-beta-ol. Now, we will see from where the cholesterol is came out and how it is used for the synthesis of different types of steroidal hormones. In first step, the cholesterol is uptake from LDL-C via LDL-R, that is called low density lipoproteins receptor, is a mosaic proteins of 839 amino acids. After the removal of 21 amino acid signal peptides that mediates the endocytosis of cholesterol rich LDL. It is a cell surface receptor that recognizes the apoproteins B100, which is embedded in the outer phospholipid layer of LDL particles. In humans, the LDL receptor protein is encoded by the LDLR gene on chromosome 19. It belongs to the low density lipoprotein receptor gene family. It is most significantly expressed in bronchial epithelial cells and adrenal glands and cortex tissues. The second way from where the cholesterol is isolated, that is de novo synthesis. De novo synthesis refers to the synthesis of complex molecules from simple molecules such as sugar or amino acids, as opposed to recycling after partial degradations. So during de novo synthesis, cholesterol is synthesized, which is used for the preparation and synthesis of different steroidal hormones. For example, nucleotides are not needed in the diet as they can be constructed from small precursor molecules such as formate and aspartate. The third way from which the cholesterol can be used for the synthesis of different steroidal hormones is cholesterol, which is cholesterol esters, or cholesterol molecules with long chain fatty acids linked to the hydroxyl group. They are much less polar than free cholesterol and appear to be the preferred form for transport in the plasma and for storage. So, the cholesterol in the first step is converted into pregnenolone by using 450-SEC enzyme. This is the first step for the synthesis of steroidal hormones, that is pregnenolone. The difference between both the structure is that there is a presence of hydroxyl group at carbon 3 positions in beta form as well as acetyl group at 17 positions in the pregnenolone structures. You can see over here some important common structures of hormones, and these hormones are cortisol, progesterone, estradiol, aldosterone, testosterone, and dihydrotestosterone. Let's see the sources, its uses, and solubility of these molecules respectively. The first molecule is cortisol. It is produced in many animals, mainly by the zona fasciculata of the adrenal cortex in the adrenal glands. It is soluble in water, ethanol, and isopropanol. Cortisol regulates a wide range of vital processes throughout the body, including metabolism and the immune response. It also has a very important role in the helping the body respond to the stress. Cortisol has a molecular formula that is C21H30O5, means there is a presence of total number of carbon atoms 21 and hydrogen 30 and total number of oxygen atoms that are 5. The molar mass of cortisol is 362.46 gram per mole. The next molecule is progesterone. When we see the structure of this molecule, we observe the presence of carbonyl group at carbon 3 position and acetoxy group at carbon 17 positions, as well as a double bond is present at carbon 4 position. The chemical formula of this molecule is carbon 21, hydrogen 30 and oxygen 2, means there is a presence of total number of carbon atoms 21 and hydrogen 30 and total number of oxygen atoms are 2, that is presence at carbon 3 and carbon 17 positions with acetoxy group. The molar mass of progesterone is 314.469 gram per mole and melting point is 126. Progesterone can be used as an administrative route, that is taken by mouth. It can also be taken as topical transdermal as well as vaginal and intramuscular injection. It can also be used as a subcutaneous injection and a subcutaneous implant. Let's see the sources of this molecule. Progesterone can be isolated from beans, broccoli, Brussels, sprouts, cabbage, cauliflower, kale, nuts, and pumpkin. This hormone is produced in the ovaries and the placenta when a woman gets pregnant and the adrenal glands. Progesterone is soluble in organic solvents such as acetonitrile, ethanol, and methanol. Progesterone prepares the endometrium for the potentials of pregnancy after ovulation. It triggers the lining to the thicken to accept a fertilized egg. It also prohibits the muscles contractions in the uterus that would cause the body to reject an egg. The next molecule is estradiol. When we see the structure of this molecule, it consists of hydroxyl group at carbon 3 position and the other hydroxyl group at carbon 17 position. The total number of rings in these molecules are 4. 3 rings are hexagonal and the fifth ring is pentagonal. However, the first ring is an aromatic. The chemical formula of estradiol is C18H24O2, means it comprises total number of carbon atoms are 18 and hydrogens are 24 and oxygen atoms are 2. The molar mass of this molecule is 272.38 gram per mole, and this molecule can be used as an administrative dosage. It can be taken as an oral, sublingual as well as intratropical, transdermal, vaginal, intramuscular, subcutaneous and subdermal implants. Estradiol is also known as E2 and also spelled with estradiol, is an estrogen steroid hormone and the major female sex hormones. It is involved in the regulations of the estrus and menstrual female reproductive cycles. Estradiol is responsible for the development of female secondary sexual characteristics such as the breasts, widening of the hips and a female associated pattern of fat distributions. Now we discuss the sources, uses and solubility of estradiol. It is produced especially within the follicles of the ovaries, but also in other tissues including the testicles, the adrenal glands, fat, liver, the breasts and the brain. Estradiol is produced in the body from cholesterol through a series of reactions and as an intermediate, as you have seen in the steroidogenesis. It can be soluble in dimethyl sulfur oxide, that is DMSO, and dimethylfuramide, that is abbreviated as DMF, and in ethanol. When we see its uses, estradiol regulates many processes in the body. Estradiol is used to treat menopause symptoms such as hot flashes and vaginal changes, and to prevent osteoporosis, bone loss in the menopausal conditions of the women. Let's see aldosterone. This molecule consists of total number of carbon atoms 21 and hydrogen atoms are 28 and oxygens are 5. There is a presence of carbonyl group at carbon 3 positions and one methyl groups at carbon 9 position, hydroxyl group at carbon 11 positions and an aldehyde group at carbon 13 position. In addition with the methoxy group which is attached to the carbonyl group at carbon 17 position. So the molar mass of this molecule is 360.450 gram per mole. Let's see the solubility, uses and sources of aldosterone. Aldosterone is produced in the cortex of adrenal gland and it is soluble in organic solvents such as ethanol, dimethyl sulfur oxide and dimethylfuramide, which should be purged with an inert gases. Aldosterone affects the body's ability to regulate blood pressure. It sends the signals to organs like the kidney and colon that can increase the amount of sodium the body sends into the bloodstream or the amount of potassium that is released in the urine. The next molecule is testosterone, which comprises of carbon 19, hydrogen 28 and oxygen 2. You can see over here in the structure of testosterone there is a presence of carbonyl group at carbon number 3 and a double bond at carbon number 4 in the first ring, in addition with the hydroxyl group at carbon 17 position. Testosterone is the primary male sex hormone and anabolic steroid. In male humans, testosterone plays a key role in the development of male reproductive tissues such as testes and prostate glands, as well as promoting secondary sexual characteristics such as increased muscles and bone mass and the growth of body hair. In addition, testosterone is involved in health and well-being and the prevention of osteoporosis. Insufficient levels of testosterone in men may lead to abnormalities including frailty and the bone loss. The chemical formula of testosterone is C19H28O2 and the molar mass is 288.431 gram per mole and melting point is 151.0 degree centigrade. Let's see the sources, uses and therapeutic of testosterone. It is produced by the man gonads, which is the major source for the production of the testosterone. Testosterone is soluble in ethanol, methanol, and it is used for primarily to treat symptoms of sexual dysfunction in men and women and hot flashes in women, in addition with to increase the desire of libido. Let's see the last molecule which is presented on this page is DHT, that is dihydrotestosterone. The molecular formula of dihydrotestosterone is carbon 19 H30O2 and molar mass is 290.447 gram per mole. So there is a difference between testosterone and dihydrotestosterone, only the presence of double bond at carbon 4 positions, which is present in testosterone and however absent in the dihydrotestosterone, while all other structural features are the same as that of the testosterone. Most DHT is produced in the peripheral tissues like the skin and the liver, whereas most circulation dihydrotestosterone originates specifically from the liver. So these are the major sources from which dihydrotestosterone is generated. Dihydrotestosterone is soluble in dimethyl sulfur oxide and ethanol, and when we see its uses, DHT is biologically important for sexual differentiation of the male genitalia during embryogenesis, maturations of the sex organs and scrotum at puberty, growth of facial, body and pubic hairs, and DHT is used for the development and maintenance of the prostate glands and seminal vesicles. Let's see the isolation and purifications of steroids. This is the specific method for the extraction and isolation of steroid molecules. In this method, we will take hot boiled egg yolk in which 1 gram of potassium carbonate is added. After that, 5 gram of sand and 10 ml of methanol is added. Whole material is grinded and we will get the soft scrambled egg material. In this material, we will add 20 ml of cyclohexane and stir it for some time. After that, we will take the cornmeal mush material. The whole material is then formed for reflex and evaporation. After evaporations, we will get the evaporated egg mixtures in which we will add 30 ml of methylene chloride. Now, This material is subjected for column chromatography, in which we will take 15 gram of flash silica gel and layer of sand on the top. So the column is packed. After that, the whole column is eluted with 30 ml of methylene chloride. Now it is rinsed with ethanol with the help of petroleum ether. After that, again the elution is carried out with the help of ethanol and petroleum ether with the specific ratio. So in this way, from the column chromatography, we will get total 15 fractions, and each fractions have 10 ml of volumes. Now each fraction is subjected for thin layer chromatography for the identifications of the pure compound as well as for the presence of steroid molecules. So the cholesterol will be spread into 6 to 16 fractions. Now after the evaporation of cholesterol fractions, we will get the cholesterol crystals in the round bottom flask. Now the steroid, which is in the form of cholesterol, is recorded for weight and then taken its melting point. After that, the cholesterol is identified with the help of specific test for the conformations of the steroid molecules. So this is the specific method for the isolations and purifications and identifications of the steroids natural products. Dear students, these are the assignments and the quiz which you would have to submit at your earliest. That is, describe the function of hormones. Also share its some nutraceutical brands with key ingredients. The second assignment is discuss the mode of mechanism of cholesterol in human physiology. Thank you.